Okay, so hello. Welcome to the session for week 8. Today we do not have any students joined as of now, but it has already been nearly 10 minutes, so I'm starting the session. The video for today's lecture is not switched on because there's some issue with my internet. If it is eventually okay, I will switch it on during the course of the class. So before we get started, there has been very little interaction on the course for the past few weeks. If there's any difficulty, if there is some feedback that can be added from your end, it would be extremely beneficial because it's a little difficult to teach or to carry out or conduct these classes without any interaction from the other side. But I understand that most of the time, a lot of doubts get resolved because of the books you study or because of whatever other accesses you might have. So irrespective of that, if some students could join, it would be extremely helpful. So anyway, with this discussion, I will now begin the discussion for week 8. Now, before we get started, to be very direct about it, we have to keep in mind that a lot of topics that were covered for this week have already been introduced in some of the previous weeks when we were discussing B cell receptors and the VDJ rearrangement, when we discussed about the organs, the secondary organs within the body, primary and secondary immune organs within the body, including the thymus and the lymph nodes and how exactly they function. So we have some concepts or some of the preliminary knowledge that you require to begin this course. But yeah, despite that, a lot more detail has been introduced in this week regarding B cell maturation, its activation, and the downstream way. The downstream pathways that allow it to execute its immune activity, apart from just the fact that antibodies are able to regulate their immediate action. So while B cells definitely constitute a major part of the immune system by contributing their receptors, which is B cell receptors or antibodies, that have their own set of cascades that they are able to trigger. There are also other pathways, which if you've already gone through the lectures, you're already familiar with. But yeah, we will be introducing it a little further in this class. So before we move ahead, we will quickly recap what this is already a slide you're familiar with. So this is a slide you're already familiar with. So here we had discussed B cell development. Wait. Yeah. So we had already discussed the overview of B cell development, how it occurs, where it is, like the localization with respect to organs, what exactly facilitates it, and so on and so forth. So if you remember, of course, there are two major organs that are participating in this. You have the bone marrow, whereby you have a progenitor B cell, which is a primitive B cell that is undergoing the IgG immunoglobulin gene rearrangement and some selection procedures that are ultimately generating the B cell. And from there, approximately, you can see that nearly 5 into 10 to the power 6 cells are produced in a day. That's approximately 5 million cells a day. And then they undergo some downstream processes, which occurs not within the bone marrow, but instead occurs within the peripheral lymphoid organ, which is considered to be the antigen dependent phase. So while the first phase is maturation, which is from the generation of a mature B cell from a progenitor cell because of some gene rearrangement pathways and some selection pathways. The second part depends upon interaction or when when a B cell is able to encounter its cognate antigen, the downstream processes that are observed there. So some parts of this we obviously already have discussed in the over the several weeks that we have discussed this course. So you see that when there is no antigen, there is cell death in approximately 90% of the cases, whereas when it encounters its cognate antigen, which is a slightly less common event, only occurs in near about 10% of the cases. It is then activated so that it can undergo affinity maturation to generate memory B cells and undergoes class switching. Then it switches to the exact it switches so that it undergoes class switching so that it can generate the exact type of antibodies which would be the most efficient or the most useful in eliminating the pathogen of our concern. and then it secretes, it can secrete those antibodies so that they can move about in circulation and clear off the infection that is encountered in the first place. So that is what we have already learned about. If we quickly go through the legend, first of all, we see that during the antigen-independent maturation phase, immunocompetent B cells expressing membrane IgM and IgD are generated in the bone marrow. This is occurring within the bone marrow itself. Of this, only about 10% reach maturity and are able to exit the bone marrow. Naive B cells, which are present in the periphery, naive B cells are those which have not encountered their antigen. They are naive B cells that have the potential to be immunologically active, which means that they are immunocompetent, but the absence or the lack of their cognate antigen is causing them to remain in their naive state, and therefore they must eventually be cleared out from the system. So naive B cells in the periphery die within a few days unless they encounter their soluble protein antigen and activated Th cells. Once activated, B cells proliferate within secondary lymphoid organs. Those bearing high affinity membrane immunoglobulins differentiate into plasma cells and memory B cells, which may express different isotypes because of class switching. This is what we just talked about. And here, of course, this was a sort of disclaimer that all the statistics you see here are based on what is observed in mouse, but the overall idea or the process or the um, components of this pathway remain the same. So with this preliminary revision of something we have already talked about, let us now move into the specifics of B cell maturation. So like you saw the second part of it, of what we saw, which takes place within the secondary or the peripheral lymphoid organs, that is an antigen dependent phase and is heavily dependent upon the initial recognition between a B cell and its cognate antigen. This part or the maturation part is something that is more preliminary, is considered to be the antigen independent phase, and its location is within the bone marrow. So our first focus today starts off there. So as you can obviously understand by the name maturation, it means it is going from an immature to a mature state. What exactly happens there? And keep in mind that you cannot really use terms very loosely. I just mentioned immature because it means it's from a non-mature state to a mature state, right? However, the terminologies that you use here should be very specific. And in this case, immature is not exactly the correct word. Why? You'll understand it very soon. Here you can see that this is showing you an example of a bone marrow stromal cell. So if we just quickly go back to this slide, yeah. So within the bone marrow, there is this process that is allowing this transition from a progenitor to a mature B cell is what we are discussing today. So what is present within the bone marrow? Of course, among other components, one major component is the presence of stromal cells. They form the stroma of the bone marrow that is present within the bone. Now, these stromal cells, they are the scaffold or the site of interaction for, or the site where multiple interactions, very productive interactions occur that allows the conversion from an image from the progenitor to the competent B cell. So what exactly happens there? So let's go with the legend and the figure at the same time. So bone marrow stromal cells are required for maturation of progenitor B cells into precursor cells. You can see that the first stepping stone involves the presence of pro B cells. This is the first stage. If you draw it like a flow chart, then the topmost stage or the step is the pro B cell. What it does, the pro B cell is able to bind to this stromal cell present within the bone marrow by means of an interaction which is present between VCAM1 and VLA4. VCAM1 is present on bone marrow stromal cell. If you've gone through the lectures, then you already know that VCAM1 is a cell adhesion molecule present on the stromal cell. It forms a very specific, very cognate interaction with VLA4, which is a leukocyte antigen that is present on these very primitive pro-B cells. So this is the first interaction. Next, from the pro-B cell to the pre-B cell, other interactions come into play. Here you see that there is the interaction which is called as which is the result of the, okay, let me reframe it. So this first interaction facilitates the presence or the interaction between two other proteins. So what it basically does is when the first interaction has taken place, then it is allowing you to go ahead, or it is allowing the cell to go ahead or even look for the second interaction. So it is the second interaction is dependent upon the first. That's why it's been put like that. So this interaction promotes the binding of c-Kit on the Probes cell to the stem cell factor, or SCF, which is present on the stromal cell. This triggers a signal which is mediated by tyrosine kinase activity of c-Kit. c-Kit is a receptor tyrosine kinase. And when this interaction is occurring between c-Kit and stem cell factor SCF on the stromal cell, it is generating a downstream signal that stimulates the Probes cell to express receptors for IL-7. So you can see that every subsequent step is dependent upon the one which comes prior to it. So yeah, so obviously we see that BLA4. What you also need to keep in mind is that, again, this is a textbook diagram, but if you've gone through the lectures, there was another reaction which occurs even before that, before this one, which is the reaction between FLT3 and FLT3 ligand. So yeah, so FLT3 and its receptor, actually, sorry, there was some issue. And the presence of that interaction is what facilitates the interaction between BLA4 and VCAM1, following which we see that there is another dependent reaction, which is between c-Kit and SCF. Yeah. And then you see that that facilitates the expression of certain receptors on the cell surface, which are called as the IL-7 receptors, or the expression of IL-7 on the surface of bone marrow cells. And that allows it to bind to the IL-7 receptor. So coming back to the legend again, the IL-7, which is released from the stromal cell, is able to bind to these IL-7 receptors, which induces this to eventually mature into the pre-B cell, and the further proliferation and differentiation eventually helps in the production of immature B cells. Now, this actually you can see has a population whereby you can see the presence of some MIGMs on its surface. And then depending upon the type of antigen that has been encountered, it can further class switch into IgG, A, M, E, and D, or in between these options. And then the most suitable one will be the one that will be allowed to persist. Yeah. Then you see this is a comparison that shows you the sub-steps or the sequential events that occur over the course of B cell maturation in the bone marrow. So you can see that there is, if you first look at the topmost schematic diagram representation, you see that there are this major division between two states. One which is yeah. So one which is a bone marrow antigen independent state, and one which is a dependent state. When we talk about the independent state, that initially begins with a lymphoid stem cell. So first is your hematopoietic stem cell, from which you get your lymphoid progenitor. If you very, quickly recollect the way your T cell development was also occurring, which is something we just discussed the day before yesterday. Then from that lymphoid stem cell, progression occurs into the pro-B cell, which then matures into the pre-B cell, following which it develops into the immature B cell, naive B cell, and mature B cell. Now, I hope you can understand that why did I say that you cannot use terms very loosely? Because if you just give it a quick look, it will appear as if all of these terms are simply interchangeable. You can use one in place of another, but that is never the case. Pro and pre-B cells are different. The surface expression markers, their jobs, the physiological changes they have individually undergone, all of that is very, very varied, and therefore needs to be, taken into consideration separately. And also, an immature B cell versus a naive B cell also has certain, very subtle, but certain differences. We'll come to it. So let's see how they actually differ. So if you look at the legend, it says that the pre-B cell, it expresses a membrane immunoglobulin or a membrane antibody that consists of a heavy chain and a surrogate light chain or surrogate light chains, which are called as V pre-B and lambda 5. You will be seeing this very soon in a schematic representation as well. Changes in the RNA processing of heavy chain transcripts following the pre-B cell stage leads to the synthesis of both membrane-bound and the secretory form, both membrane-bound IgM and IgD by mature B cells. So if you remember again, the VDJ recombination classes that we had like a few weeks ago, then you'd remember that the first antibodies that are produced on cell surface are these, and then depending upon the type of antibody and the type of specificity that is required, it will change. Or the type of parasite that it basically encounters, it can change into the more efficient subtype of an antibody. Yeah. What you need to remember as part of the legend also here is that RAG1 and 2 have been indicated. RAG1, RAG2 are two enzymes that are encoded by recombination activating genes, which is what RAG stands for. TDD is for terminal deoxyribonucleotide transferase. And also you can see that a number of B cell associated transcription factors are very important at different stages, some of which have been shown here. So let's take it one at a time. First, if you see the lymphoid stem cell, what are the characteristic features of this particular cell? Its heavy and light chain genes are as observed in germline. It has obviously not undergone any rearrangement. Yeah. However, if you see the pro and pre-B cells, they have some difference in their properties. Compared to the lymphoid stem cell, in the pro-B cell, you see that there is some heavy chain gene rearrangement that has occurred, leading to the generation of DHJH. Basically, there has been DJ joining, not VDJ, but just the D and J regions have undergone some recombination, and they have, they have combined with each other. When we talk about the L chain genes or the light chain genes, however, you see that you don't have a functional or an actual light chain, rearranged light chain available at that point of time. But instead, these cells have a surrogate light chain, which is said to be B pre-B and lambda 5, which is germline in nature. In comparison, some disruption or something. However, in comparison, you can see that within the pre-B cell, there has been VD and J recombination. All three of them are present, and it is accompanied by the same surrogate light chain. Once this VDJ recombination has been performed, that will be carried, and that does not undergo very significant changes except some minor, editing to some extent. What you also now need to understand is that there's a very, very huge difference between editing of a gene and rearrangement of a gene. Editing is something which is occurring at a very individual base level, that there is a change in one or two bases, or very, very minimal changes are occurring. However, when you talk about something like a rearrangement, it means that there has been removal of a stretch of nucleotides or a stretch of DNA. Let's put it like that, a stretch of DNA, and therefore it is shifting completely from one side to another. With that possibility, the event, such an event which is so energy consuming and is such and can have such huge impacts on, cell survival at large, you would obviously need to understand in that case that once rearrangement has been finalized, it is maintained, and only very subtle changes may or may not be required or acquired over the course of time. So therefore, VDJ recombination for heavy chain is being maintained throughout. However, the rearrangement of light chain genes is occurring in the next step, which is for immature B cell, and then that rearrangement is also being carried throughout the life of those cells, and is consistently being maintained across cell divisions as well. Next, you see the presence of the enzymes RAG1, 2 and TDD. RAG1 and 2 are only present in the pro-B cell and pre-B cell stage. That is, this is a recombination associated gene, and therefore its expression is extremely crucial. Say, for example, if there was never a check on how much of the RAG1 and RAG2 is being expressed, is it functional or not, how long is it persisting. If nothing like that was done, it could eventually end up causing havoc within the cellular genome and could potentially lead to very severe diseases as well. However, its expression is therefore very tightly regulated, and you only see its presence in a sort of localized manner at the regions where rearrangement is crucial, that is at the VDJ um, sites. So with this under consideration, you can see that there are only two states or two conditions whereby RAG1 and 2 are not available, and that is during the pro-B cell and pre-B cell stages. Following this, we see the terminal deoxynucleotide transferase as well, whose expression is exclusively only limited to the pro-B cell, which is the more primitive condition. Yeah. Next categories or next set of categories that we can look at are those that involve membrane Ig heavy chain and light chain as well. You can see that in the pre-B cell, you can see the expression of IgM, which is the preliminary heavy chain that is expressed and is present on the cell surface in combination with a surrogate light chain as well. However, that undergoes class switching and can eventually go and develop into the into a or can eventually inculcate the expression of both mu and delta, which is IgM and IgD molecules on the cell surface as well. On the other hand, if you talk about the light chain, then only kappa or lambda would be expressed, and that occurs after VJ recombination for the light chain gene in the immature B cell. In contrast, the transcription factors that are expressed in each condition or each cell type are extremely varied, as you can obviously see. While you see that the lymphoid, in fact, this might be very useful information for you as well, not only with respect to, this course and just getting through assignments or clearing the exam, but instead it would be very, very useful if you want to understand how, like if you're interested in stem cell biology or for that matter, even cancer biology or immunology in the long run, and you want to understand how stem cells can actually be identified. Why I'm saying this, because once you have theoretical knowledge, you would be able to understand and even hypothesize or speculate how they can be practically applied. Say, for example, we have learned about FACS in just as part of this course, right? Course, which is fluorescence assisted cell sorting. So FACS and flow cytometry are an immensely used, very well-studied technique in context of molecular biology. And you can see very well that the reason that they are able to perform this for different cell types is because of the presence of certain surface markers or presence of some differences between these cell types on their surface. Such an example, when, for example, you want to separate a stem cell population from the non-stem cell population, is the presence of surface markers like this, one of which is c-KIT. So c-KIT is a cell surface marker, but also you need to understand they are not very, very abundant on the cell. There are very few c-KIT molecules which are present on the surface. But yes, it's definitely a stem cell marker. And there are also other markers as well, like CD44, etc. So using antibodies specific for these antigens or these surface molecules can allow you to segregate the different subpopulations which are present within the um, within your sample which you are trying to analyze. Next, you see that transcription factors for stem cells, lymphoid stem cells are PU.1, Icaros, etc. However, for pro-B cell and all the other downstream transcription factors that you can see, there is a shift and there's a large scale expression of several downstream genes. Starting off with examples like BSAP, SOX4, E2A, OCT2, EBF, etc. These are being maintained throughout and they're necessary for their proper immunological and physiological functions as well, for them to grow, proliferate, and expand the repertoire of cells that is present. And we talked about the surface marker of the stem cell, but for the subsequent cell types, you can see you have different markers. Most understandably, most useful marker in this case would be the presence of IL-7 receptor, which is present here, because as you remember in the diagram we just saw just a little while back, that after the initial interactions between VLA and VCAM are facilitated, and that is accompanied by another set of interactions. Just let me go back here. Yeah, another set of interactions between c-Kit and SCF that allowed the induction or the release of IL-7 from the stromal cell, which was able to bind to the IL-7 receptor that is present on these different pre-B cells, and then they can undergo maturation. So therefore, its expression is also definitely on the surface. So that can be used for distinguishing it as well. So basically, yeah, this is a lot of just fact-based memorizing zone. Like these are factors that you just have to remember, very simply put. But yeah, it's useful, and like I mentioned, its practical application is what matters. Just knowing that a stem cell has a particular marker on its surface will never do you any good. What you need to understand is how does it matter? What if I remove that gene? What if I increase that gene? Can I just simply introduce it to a non-stem cell population, and will it start behaving like a stem cell? Or if I withdraw or inhibit the expression of that gene, will it affect the function of the original cell as a stem cell? So these are the type of questions that you need to understand, you need to address, not only with respect to stem cell genes, but for that factor, any gene at all, whenever you want to uh, you know, um, understand its physiological role within a system. So yeah, just a second. Yeah. Now, proceeding forward, you can see in a little more depth about the interactions which are occurring during B cell maturation. So there's some issue with Okay. Anyway, what you see here is the sequential expression of membrane immunoglobulins and the associated surrogate light chain that is present at different stages of B cell differentiation in the bone marrow. The pre-B cell receptor contains a surrogate light chain consisting of a V-pre-B polypeptide and a lambda 5 polypeptide. If you remember, we had just discussed this, I think, in the previous slide. So basically what you see here is that in the first condition, you have the pro-B cell, which is expressing Ig alpha beta receptors on the cell surface. Next, you see a pre-B cell, which has undergone to some extent, it has undergone uh, some changes or some modifications, and it has progressed towards, or it has transitioned towards the pre-B cell. Now, what happens with respect to the pre-B cell is that it contains the surrogate chain that is consisting of some V pre-B polypeptide and another lambda 5 polypeptide, which are non-covalently, in this case, which are non-covalently being attached. Yeah, so what you see here is in the blue sort of cylindrical structure is the heavy chain, whereas not cylindrical structure, but the blue lines basically are the heavy chain, whereas you can see the light chains to be associated with it. And in this stage or at this stage, it is not a regular light chain, or it's not a complete light chain, but it serves as a surrogate light chain just to, sort of maintain the structure of the antibody until it can bind to its cognate receptor. Not receptor, but is able to combine with its correct light chain. Let me just put it that way. Also, within the surrogate light chain, you see within the diagram that there are two subtypes, and they have been identified in this case as V pre-B and also the lambda 5 subtype. Hello. I'm not exactly sure if my audio is working, but okay, anyway, we'll just go ahead with it now, and I am hopeful that it's working properly. So, so you can see that what it basically does is it stops VH to DHJ joining, which is allelic exclusion, and instead it allows for the induction of VK-JK joining. So that is the pre-B cell. Following this, you have a further more progressed sort of version, which is an immature B cell, which is no longer pro or pre, but instead has moved forward and is now in a state which is not yet mature, but is very, very immunocompetent. So you have BDJC, which is present on the heavy chain, and you are able to, or you should be able to combine it with another one, which is another light chain, which is either of kappa type or of lambda type, and then you have the entire antibody, which is basically being formed. And once such a receptor, which is present on an immature or a naive B cell that has on a naive B cell, just put it that way, then what it effectively does is it can lead to one of two possibilities. Either it will trigger death or it will trigger activation. When we say it will trigger activation, it means that if that interaction upon after you have the knife cell, if it is able to find its cognate antigen, then it will certainly be selected for, and that will become its positive selection sort of, and then that will expand the numbers. Whereas the other possibility is that if that is, leading to a condition where it is, where this antibody is able to bind to a self antigen, then in that case, it would lead to very serious autoimmune disorders. And therefore, in such cases, they are eventually subject to death. Uh, yeah. Now, this actually diagram, it shows you a schematic representation as an evidence for negative selection, otherwise what is called as clonal deletion. Yeah. So negative selection or clonal deletion can be performed in this method. As part of it, what we see is that the yeah the presence or the absence of mature peripheral B cells that express a transgene encoding IgM against the class 2 MHC molecule K kappa was determined in H2 alpha K and H2 delta mice. In the H2 decade or delta kappa transgenics, the immature B cells which are present, they recognized or they were able to recognize K uh, and were deleted by negative selection. So basically what it means that if you have a transgenic mouse, which has the genotype alpha and K, which means it has been generated as a combination between two mice, one of which had the strain alpha, the other had the strain K, and therefore the F1 generation of such a mouse, or just the mm, offsprings of such mice, would be transgenic in nature, and they would have some attributes from both the participating parents. Uh, Yeah, here we are saying that in this H2 alpha K transgenics, first what happens is a clearance for any and every B cell which is able to bind to its own antigen. Why so? If it does so, then it will, remove that antigen, and even if it is crucial for the cells will not necessarily always be able to fight it off. Um, yeah, so that is the first condition on the left in the figure, that it is a strain of mouse, which is a transgene, which has a transgene for D and K. Yeah, D and K. And what you see in such cases is that no mature B cells can express anti-KK. Second hypothesis or the second strategy is that there are H2D transgenics which can be performed, and Yeah, on the cell surface, you are able to see KD receptors, which are present in marked in red, and they are claiming that nearly 25 to 50% of mature B cells are able to express this anti-K. More detailed analysis will reveal even more peripheral B cells, and basically that's how it works. Now, the third possibility is where it gets interesting. Um, okay, so apart from the fact that Yani, apart from the fact that there is some extent of gene editing which is allowed, uh, replacement is not really allowed. So just you are editing or changing a few bases, which can, alter its eventual specificity as well. Yeah, so we have H2 DK transgenics, which are undergoing to some extent some, light chain editing. And what we eventually end up getting are a few mature B cells with new light chains that are no longer able to bind to that particular condition. So we had, like apart from this also, if you have, carefully studied the lectures for this week, then you'll see how Professor has explained all of the different possibilities as well. So either if it is very directly, if we call something, if there is a, you know, if your system has a self antigen to which a particular antibody can bound, or such a particular T cell will have to be eliminated because that can attack the body's own cells. That is certainly not should not be the case. Um, Second possibility is that if there is a very, you know, yeah, okay, if there is to some extent you see some interactions which are occurring, then in that case, you should uh, you should be able to tighten out how many cells have actually been able to, go through this entire maturation and selection process. And third, yeah, for this. But in the third possibility, you can see that there can be to a large extent, you can edit the light chain. And as a result, you will have a sub population that behaves slightly, hopefully very slightly different. Okay, just give me 2 minutes. I will be back. Okay. Now in the heading I've written T cell antigens, but the objective was to now bring about the substeps that come in B cell activation. What we saw before this was the maturation of B cells. I'll change the title just to avoid any, discrepancy in this. But yeah, here you saw the maturation of B cells. Now talking about the activation part. Yeah, so when we talk about B cell activation, it is obviously mediated by the antigen, but there are also other participants that help in execution of this job smoothly. Again, if you have listened to the lectures and studied from the lectures already, you would be familiar with it. But in case you have not, this would be a slightly new concept and a very interesting one at that as well. You will see, okay, let me first explain it, then I'll tell you what to, uh, as a probably as an aspiring researcher, if you eventually want to, go ahead with this line, what should be the way of you conducting your critical thinking or, cultivating your critical thinking skills. So we'll come to that. But yeah, basically what you see here is that for B cell activation, an effective signal, it involves two very distinct signals um, that are induced by certain events that are occurring right at the cell membrane. So you see that binding of a type 1 thymus independent antigen to a B cell provides both signals, signal 1 and signal 2. A thymus-dependent antigen provides signal 1 by cross-linking membrane Ig or membrane immunoglobulins. How is that executed? Because of the binding of the antigen to antibodies present on the surface are able to cross-link. And yeah, but a separate interaction with or between CD40 and CD40 on the B cell and CD40 ligand on an activated TH cell is necessary to generate the second signal. Yeah. So what you see here is that you have a B cell, and at its cell surface, you can see two schematic representations of antibodies which are able to bind to their cognate antigen and then activate the downstream signaling pathway. However, you can see that when we talk about a tumor-dependent antigen, things are different. So you have a B cell. Yeah, you have a T cell dependent antigen. So your B cell, while it is interacting with that particular antigen, it is also receiving in parallel, it is receiving stimulus or rather, stimuli from an adjacent or a neighboring T helper cell, and there are physical contacts that are made between these two cells for this purpose. Why did I, specifically stress on the fact that there must be some physical contact between the T cell and the B cell for it to be considered a thymus-dependent response? Is because when we actually characterize the independent subtype or the T cell independent subtype, then in that particular characterization or in that particular group, you do not have physical contacts, but there is a subgroup which is actually inducible, which is actually inducible by some factors that are synthesized by these cells. So for example, So for example, say you have some cytokines or some chemokines which are being released, and while they will certainly execute their paracrine action and will act on other cells, um, yeah, thereby making it, extremely useful. You can also see that it is not making any physical contacts, and therefore it's the signaling pathways that are activated by it and its overall effect on the cell would be quite different. So depending on the nature of the antigen, B cell activation proceeds by two different routes. I'm reading out the legend. One, which, like we discussed, is dependent upon these T cells, particularly the T helper population of cells, and the others that do not. The B cell response to thymus dependent antigens requires direct contact with TH cells, not just an exposure or not simply an exposure to the TH cell derived cytokines. Antigens that can activate B cells in the absence of this kind of direct participation by T cells are known as thymus independent antigens. Now, thymus independent antigens are getting further divided into subtypes 1 and 2, and then they are able to activate B cells by many different mechanisms. Some bacterial cell wall components Yeah, some bacterial cell wall components, including lipopolysaccharides, are able to, or LPS, are able to function as type 1 antigens. Type 2, no, TI2, which is actually thymus independent second type of antigens. These are capable of recognizing highly repetitive moieties or highly repetitive molecules such as polymeric proteins or other polysaccharides that we don't exactly understand at this moment. So basically what we see is that there are sub-classifications within the thymus dependent and independent as well. And depending upon those subclasses also, you would be able to see their specificity for different types of molecules. So yeah, so with this understanding, let us now look at what the table is basically showing to us. So you can see that there are different or classification has been performed based on certain properties. So you have based on the chemical nature, you can see that T thymus dependent TD versus TI or independent are being compared. So the chemical nature of the antigens that can be bound in case of for TD antigens, you would have, say, for example, soluble proteins. Not example, but what it basically means are soluble proteins. Um, Type 1 is for bacterial cell wall components, LPS, while on the other hand, we have type 2, which has polymeric protein antigens and capsular polysaccharides, etc. Next, we see humoral response comparison between these: isotype switching, affinity maturation, immunologic memory, and polyclonal activation. So you can see that among these, the TD antigens would be soluble in most cases, except for physiological activation. Um, yeah. Polyclonal activation or physiological polyclonal activation. And for TI antigens or thymus independent antigens, you can see two subtypes, which are type 1 and type 2. And for type 1, the bacterial LPS or the lipopolysaccharide moiety, which is present on the cell surface, which forms a very, very major component of the bacterial cell wall, that is getting recognized by the subtype 1 of the thymus independent antigens. Yeah. On the other hand, you can see that for type 2, they are polymeric protein antigens and other capsular polysaccharides, for example. Yeah. So when we talk about the humoral response, thymus-dependent antigens are able to undergo all of these processes, while thymus-independent antigens do not undergo or do not trigger uh, the following behaviors, and therefore they are not. They're different, not compromised, but it's different. Now, when we talk about the B cell mediated signaling, so what do we know till now? You have your, say, antigen X, which is able to bind to some receptor X as well, or X receptor as well. And then what it effectively does is that it is transducing some signal. Like we see that, yeah, we see that there is activation, but to understand what are the molecular mechanisms that are helping the activation, we can unravel a little more detail about this B cell response overall. So, referring to the legend, I'll just give a brief overview of this as well. So, you see that the initial stages of signal transduction by an activated B cell receptor are as follows. So, the BCR comprises an antigen binding membrane immunoglobulin and one signal transducing heterodimer, which is Ig alpha, Ig beta. And you can see how closely they are associated with respect to each other, so that see, the basic understanding is that physiologically molecules are not being brought towards each other by by hand. They are all present inside the cell at different locations. Some are soluble, means that they're simply present in solution. Some are bound on some membrane, on some cellular membrane, in most cases, or they're living as a virus, or some sort of an availability or some sort of an interaction occurs. Now, to intensity of that interaction, so as to increase the feasibility of that actually occurring, these two molecules must be brought very close to each other. Now, that is particularly relevant because, as you can see, antigens can only bind on a cell surface receptor. But if you again look at the structure, you would see that its cytoplasmic tail is unfortunately quite short. As a result, such receptors are not always very efficient at transducing signals. They are very good at um, sensing whether their ligand is present or not, and therefore they are doing their job as a receptor, but their downstream activity efficiency is always debatable. Therefore, such proteins, they are usually found to associate with some mediators of these interactions, which help in facilitating or sort of smoothing out the gaps, and therefore they're quite useful. Let's put it just that way. Okay. So following antigen cross-linkage of the B cell receptor, you can see that these are two adjacent antibodies very close to each other. Once the antigen binds It binds in such a manner that closely situated antibodies would be able to or would get linked to each other. And this is called as the cross-linking of these antibodies. Why is that relevant, you may ask? That is relevant because it's sort of like a preliminary signal that a functional interaction has happened, and now you get prepared to generate a response for it. So, it's sort of a the first call or the first indication that B cell response is about to be elicited. So you can see that these two antigens will get crossed to each other. These two antibodies will get cross-linked to each other via these antigens. And what that effectively does is that the associated uh, Ig alpha beta receptor moiety, which you can see, had some downstream motifs. And these motifs are called as ITAMs. You have already learned a little bit about them in the official classes for this week. But what they effectively end up doing is that they are motifs that are capable of undergoing phosphorylation. They're called as immunoreceptor tyrosine-based activation motifs. Um, yeah, so or ITAMs as they are commonly understood. And here you can see the sequence of the ITAM as well. And therefore, they're very useful. So, following an antigen cross-linking, the immunoreceptor tyrosine-based activation motifs interact with several members of the SRC family of genes as well, of tyrosine kinases as well, activating the kinase. The activated enzymes are then able to phosphorylate tyrosine residues on the cytoplasmic tail of the Ig alpha beta heterodimers. So that phosphorylation is again a scaffolding region for the docking of other proteins. This creates a dock for the SART family of kinases and the SIC kinases, which also become activated. So that's what you see, that antigen is coming. It is cross-linking to antibodies, which is causing the downstream pathway to be, or the downstream signaling cascade to be activated. Yeah. So, sorry for the disruption. But yeah, you can see that the highly conserved sequence motif is shown with tyrosine residues. And this is where the phosphorylation occurs, and that creates the docking site for the tyrosine receptor kinases, which basically means that these are not kinases, but these are proteins which are able to bind to the phosphorylated state of tyrosine residues and then execute its action downstream. Now we'll just go through this process. I have the summary of the entire process and the nitty-gritties of what is happening inside the cell at this point of time. So you have the compartmentalization of function within receptor subunits. Both B cell and T cell pathways begin with antigenic receptors that are composed of an antigen binding and a signaling unit. Like I just said, that for a large number of cell surface receptors, usually they have a very long transduction domain as well, which means that whatever signal they are receiving, they have to conduct it or push it forward, carry it forward. Now, both the B cell and T cell pathways begin with antigen receptors that are composed of these two subunits. The antigen binding unit naturally is responsible for conferring the specificity. Specificity, but it has cytoplasmic tails that are way too short to actually be very useful at transducing signals towards the cellular cytoplasm and even if necessary further throughout the cell as well. The signaling unit that has is a signaling unit that we actually end up studying has long cytoplasmic tails that are signal transducers of the receptor complex. Yeah. Second is the activation by membrane-associated SARC protein tyrosine kinases. The receptor-associated PTKs or protein tyrosine kinases, which in case of T cells is LCK. If you remember, we had seen it in the last class. And in case of B cells, it is mostly the Blick, Lin, or Fin kinases. They are just names of the kinases based on where they have been derived from or how they were discovered. So based on that, you have different types of subfamily of kinases present, and they are able to catalyze phosphorylations during the early stages of signal transduction, which is very necessary for the formation of functional receptor signaling complex. Third is the assembly of a large signaling complex with protein tyrosine kinase activity. Now, the phosphorylated tyrosines in the ITAPs of the BCR and TCR provide the docking sites for molecules that endow these receptors, the receptor activity. Yeah. Then you have the recruitment of other signal transduction pathways. So signals that are from the BCR and from the TCR, they are resulting in the production of some secondary messengers. When I say secondary messengers, they are majorly IP3 and DAC. If you remember, we have discussed this in the previous class as well, that there are some chemical messengers inside the cell that are able to transduce or carry forward the information which is generated or which is perceived by the cell at the surface. IP3 causes a release of calcium from intracellular stores. Again, we have seen that as a well-studied policy. And DAG activates protein kinase C. A third component set of signaling pathways are those that are governed by the small protein GRAS. And also RAC, which get activated by certain signals received through the B cell receptor or the T cell receptor, PCR or TCR. One of the most important outcomes of the signal transduction process set in motion with the engagement of BCR or TCR is the generation or the translocation to the nucleus of active transcription factors that stimulate or inhibit the transcription of specific genes. So overall, what you see as major changes within this B cell mediated signaling pathway, A is that you can see that receptor subunits are now functioning in a very compartmentalized manner, very divided or very clearly distributed work. And second, you have the activation by membrane-associated protein tyrosine kinases. Third is the assembly of a large complex that has this kind of tyrosine kinase activity at large. Yeah. Then you have this recruitment of other signal transduction pathways. So signals from BCR and TCR result in the production of secondary messengers, like we just discussed, IP3 and DAG majorly. What IP3 does is it affects the cellular storage of calcium overall. Yeah, or the intracellular storage of calcium overall, and DAG or diacylglycerol, it is responsible for activating protein kinase C. You can actually study this. And finally, the last change that is observed, or that is part of the signal transduction system, is the change in gene expression. So all taken together, you can say that failures in this system, therefore, can have very, very severe consequences for such an immune system. One example of this is actually the disease, which is quite extensive, which is a disease of a lot of interest, and it's called as X-linked agammaglobulinemia. So yeah, and the clinical focus on this disease at present describes the effect of defective signal transduction on B-cell development. You can actually read up more on this disease from Kubi or for the internet for that matter, so that you understand how it functions, why is it relevant, what is the consequence of this. Now, when we talk about, just a second. Just excuse me a moment. Yeah. Now we talk about the fact that how is B cell response coordinating with the T cell response to finally, mediate the B cell effector immunity. So of course, keep in mind again that we are not focusing a lot on the humoral branch right now. We are focusing more on the fact that how is B cell doing its job. We learned its structure, we learned its physiological evolution over a period of time. We learned the difference between gene expression in each of these stages and how that is relevant as well. So first of all, you can see that if you look at it stepwise, you can see that there is antigen cross-linking the membrane immunoglobulin generating signal 1. We have already discussed this extensively. You also have, this actually leads to increased expression of class 2 MHC and the co-stimulatory molecule known as B27. So class 2 MHC expression goes up because it needs to, prepare itself for antigen presentation. And also you have a co-stimulatory molecule known as CD28, which will be able to interact with B7 and then would mediate or would strengthen the interaction further. Antigen antibody complexes get internalized by receptor mediated endocytosis and then they get degraded into peptides, some of which can actually be then loaded onto the class 2 MHC molecule because it's intracellular in nature at this point of time and then can be presented on the surface of B cells. And then that can, eventually trigger the MHC. It can lead to more and more, it can lead to a heightened immune response against that pathogen of concern for the cell for an extended period of time. Next, what you see is that Th cells, which are T helper cells. Again, the subtypes are quite different, but yeah, we have T helper cells. If you remember, 3 different subdivisions were shown by Professor Agnew in his last lecture, where he stressed a lot on the effector T cell mechanisms for our knowledge. And you saw how each type, that is Th1, Th2, and the Treg cells and what is their job, how they play a role in terms of immunity, how they have subtle differences between their immunological role and how they are actually regulated by other factors which influence its job as well. So here you can see that when we talk about particularly about T helper cells, then there is a co-stimulatory signal which is further enhancing the strength of this interaction that is between B7 and CD28 molecules, which is helping it a little forward. Then you have the third step where you can see that it is occurring. So also keep in mind that this is getting synced with the cell cycle as well. You can see that this is all occurring in the quiescent or the G0 phase basically. Yeah, this is all occurring within the G0 phase till here, and then you can see the introduction of the G1 phase, S and the M phase. So once TH cell, after these first and second interactions have occurred, which is the first signal and the co-stimulatory signal between B7 and CD28 have both been recognized. Following that, you see that the T helper cell begins to express CD40 ligand. Now, CD40 molecule is present on the surface of the cell, the B cell, whereas the CD40 ligand is present on the T cell. Their interaction is acting as the main signal, signal 2. You had the first signal, which is the peptide MHC interaction. The second step is a co-stimulatory signal, but it's not actually considered a main signal. The main signal in this case, the second main signal, is coming forward at this step, which is between B7 CD28 interaction. They provide co-stimulation to the T helper cell. Third, you can see that B cell begins to express receptors for various cytokines. So, just yeah. So they begin to express receptors for multiple other cytokines as well. So that is acting as a further, positive regulatory signaling, not a cascade, but another positive uh, regulation also, which is further aiding the selection for this particular cell population and its increase also in the number. Yeah. So you can see then that it further undergoes in the S phase, it undergoes divisions, or it increases the amount of nucleic acid that such a cell has. And then during the M phase, which is the mitotic phase, it then divides and produces more and more cells, thereby expanding the B cell repertoire. So with this, you can see you get more and more proliferation, and then those cells can, undergo specialization as memory B cells or plasma cells, and further uh, expansion on this knowledge is observed. Finally, okay, so till here, if we just go over the steps that we have seen till now, first of all, you understood the steps of, or we explained, or we went through the steps of B cell maturation, which is occurring within the bone marrow, the different substeps that are a part of it, what is helping it further. Then we went through the steps of B cell activation, the signaling pathways that support it or that aid it. And we are finally ending the steps with B cell proliferation. So you have interactions. So while this cannot be discussed in a lot of detail immediately, I'll tell you why. Because we have not yet introduced cytokines in a lot of detail. And as you can obviously understand, this interaction is very heavily dependent upon, not even this interaction, but any immune cell specialization is very heavily dependent upon cytokines in general. So how they work, again, but you need to understand the fact that they are cytokines are simply some small molecules which are mediating communication between different cells. So it will majorly be dependent on how well you can remember what they do, which cytokine does what. So it is kind of a fact-based uh, area of study. It's not very strongly something you can conceptualize, but yeah, you can understand what is the downstream effect of that cytokine doing its own job. That's something we'll cover eventually. But yeah, what you see is that you start off with an activated B cell or a centroblast. If you remember, we had discussed all of this in a lot of detail during our initial classes when we understood how B cell development is occurring. And we had talked about the dark zone and the light zone. We had talked about how there are multiple steps within which the B cells selection is being carried out. That there is selection for affinity. It should be very capable of binding to its own binding to the cognate antigen with very high affinity, but should not bind to its own antigen at all. So all of those steps were taking place, and that is what was eventually ending, not ending, but was coming to the centroblast stage. Now, from that centroblast stage, you can see that there are multiple proliferation cytokines which are coming in. Again, like I said, cytokines are simply small molecules that are helping you in in communicating information between cells. When we call something as a proliferation cytokine, it's basically because its binding to the receptor is stimulating some downstream pathway which is supporting the proliferation or division of cells. So under that, we have IL-2, IL-4, and IL-5. So those are proliferation cytokines, and from there you can see that from that single activated B cell, you are able to generate some proliferating B cells or centrocytes. Once they have proliferated and expanded in number, a significant population will then undergo differentiation and will form their differentiation towards their destined fate. Now, why one thing that you need to keep in mind is that proliferation and differentiation are, okay, this is a slightly advanced concept, but yeah, proliferation and differentiation often they are used very closely, but you need to understand that the processes are very different and are almost always mutually exclusive. So a cell, one particular cell, which is increasing in number, which is undergoing very rapid divisions, will completely opt out of differentiation under cancerous conditions. Why? Because any cell that is differentiated and differentiated cannot divide. Let's say, for example, neurons. neurons are undifferentiated, and therefore, unless they are stimulated for some reasons, they will not normally divide, and therefore what you can see is that interneuron connections can be made or synapses can be formed because of every single new memory that you encounter in the course of your life. But new neuron formation is actually extremely tough. So yeah, so these are differentiation cytokines which are coming in after proliferating T cells have been formed. And what we see as examples there include IL-2, IL-4, again IL-2, IL-4, IL-5 as well, along with IFN gamma and TGF beta. Now you might say that why are they still including IL-2, 4 and 5 while we have these others present. So it actually depends upon what state it is in. So in the first case, the initial cell that you were looking at was a centroblast. It was just an activated B cell, but it was not in that proliferation. It was not proliferating at that point of time. The binding of the cytokine triggered its proliferation. In the second stage, when you have this centrocyte, these cells are already proliferating, and when such a cell is taken into consideration, when you have another set of cytokines, differentiation cytokines bind to such a cell, it will then undergo differentiation and form plasma cells and memory cells that you can see. Furthermore, you can also see that each of these can also differentiate into different subtypes based on the type of cytokines that signal certain messages to them. Say, for example, you can see that some cells will progress towards Ig or will undergo class switching towards IgM, some will go towards IgE, some will go towards IgG, etc. So that also is a differentiating point, and that depends upon the type of cytokine which has come in and bound. And because of its binding, it has been stimulated to proceed towards one particular direction. So yeah, with this, I think I'm done for this with the slides for today. I will now just go over some questions very quickly. Just yeah. So these are some PYQs for this week, and just to get you familiarized with what needs to be understood for now. So first of all, let's go through question one. Here we're asked about stromal cells that are present in the bone marrow, and they are required for the maturation of B cells. The question is, which of the following functions of stromal cells actually help in the maturation process of B cells? First, A, that they interact, stromal cells interact with pro-B cells via VCAM1 ligand on the stromal cells. If you remember the first diagram that we saw today, this statement is correct. Second, stromal cells secrete IL-7 that induces some common lymphoid progenitors to mature into pro-B cells. So that is completing the circuitry. So once this has been stimulated, the IL-7 that is synthesized by these cells is further triggering the formation of more and more pro-B cells so that they can undergo maturation as well. So that is also a true statement. Third is that the receptor tyrosine kinase C KIT, otherwise known as CD117, on the surface of the pro-B cell, CD117 surface on the surface of the pro-B cell binds to the stem cell factor SCF present on the stromal cell. D, all of the above, or E, both A and C. So obviously, since we have already discussed these options extensively, it is actually going to be the correct options in this case are actually A, B, and C. Therefore, option D is the correct option in this case. All three are correct. Next, you have during B cell maturation, which of the following stage in which of the following stages the surrogate light chain is complex with heavy chain, and surrogate light chain which is complex with a heavy chain starts expressing. So that is actually in the pre-B cell stage. If you remember, the pro-B was the first participant or first component of the pathway, and right after it we had the pre-B cell where expression of this heavy chain is facilitated with the help of a supporting surrogate light chain. Then we have question 3, where we are asked that which of the following fates may occur to a B cell that reacts to a self-antigen. So when we talk about a self-reacting V, if you remember, again, we have discussed this extensively, that there are two types of selection. I did not segregate it very extensively today in my lecture because in my discussion, because it has already been elaborated and discussed multiple times in the already uploaded video lectures by the professors. But yeah, basically what you need to understand is that there are two types of selection pathways that operate before a B cell is allowed into circulation. One is a positive selection, one is a negative selection. When we talk about positive selection, it occurs in such a way that only those B cells which are able to you know which are capable of interacting with some antigens can be allowed to proceed, and they would be that have high affinity for the antigen that you are um, interested in, or that needs to be eliminated from the system. Only those are allowed to proceed. And negative selection is to get rid of all those B cells that are reacting with very high affinity to our own body cell molecules, or body cell surface markers, or polypeptides, let's put it that way. So both of these together are creating a subpopulation of cells that are, very specific, and that are only ensure that ensure that only external moieties are getting removed, but cellular compartments which within an individual's body are not getting compromised. But again, you need to keep in mind that this is an idealistic situation where it is not happening, and you will find a huge range of diseases where this actually happens, and you can altogether group them uh, into autoimmune disorders, which means that this is a condition where your immune system is itself attacking your own body cells. So instead of protecting it, basically what it does is it starts chewing up your own cells, not chewing up, but starts eliminating your own cells, and therefore it affects the health of an individual. And these are, as you can obviously understand, it's a serious condition, but yeah, not all of them are necessarily always lethal. There are cures to it, if not cure, at least some of them are very they can be managed with some lifestyle changes and with medicines, etc. They can be managed, but certainly they have their own set of issues that they come with. So yeah, coming back to the question at hand, the which of the following fates may occur to a B cell that reacts to a self-antigen? A, receptor editing, apoptosis, anergic B cells, or all of the above. So anergy is something that you have been already introduced to. Anergy is basically a condition where those cells will become non-responsive. So B cells that have okay, this is something that is discussed in the lecture, just as a preliminary disclaimer. That's why I'm coming to the answer directly. So first is that we saw that if it reacts with a self-antigen, then A, if it is a very strong interaction and it cannot be managed, then it will be subjected to apoptosis. Second, if it is something that some few changes or few edits can affect that um, binding specificity, then in such a case, such a B cell will undergo receptor editing and will remain in circulation. Third is a condition where if it reacts extremely strongly but is not undergoing apoptosis, or it has high affinity for some self-antigen, then in such a case, it will be subjected to anergy. Basically, it will be suppressed and its activity will no longer be functional. It will not be able to bind to its soluble antigen within the body. And these, all of the above. So all three of all the first three options are correct, and therefore the correct answer in this case is option D. Next, we have the interaction of a naive T cell with an APC induces the expression of both IL-2 and IL-2 receptor on naive T cells. IL-2 binds to IL-2R on the same T cells to induce their proliferation. Paracrine, autocrine, endocrine, or exocrine function. So I think you're already familiar with it, but let's just quickly brush up the concept. So autocrine is when a cell is mm, when the chemical entities secreted by a cell are binding to receptors. Okay, let's just use the terms directly. When the ligand released by a cell is binding to its own receptor, then that is autocrine. when such a ligand is binding to the receptor present on a different cell or a neighboring cell, then that is paracrine. However, exocrine and endocrine are completely different terms. Endocrine glands are those glands, or endocrine function is usually associated with glands because of the fact that they are able to release hormones. And endocrine and exocrine are working in different ways. Either they will release hormones that are able to act, secrete hormones that will be completely moved into circulation. Ductless glands, they're basically ductless glands, whereas exocrine glands would release them into completely into circulation. So exocrine and endocrine are certainly not applicable here. Here we are trying to understand that when IL-2, the ligand, binds to ILTR on the same T cells to induce their proliferation, then that is an example of what? So obviously that is an example of an autocrine signaling or autocrine function because it is stimulating its own proliferation by binding to a ligand that it itself had secreted. So it is kind of a you can say a positive feedback loop. Yeah. Then you have centroblasts. They move to the light zone of the germinal center because CXCR5 shows high affinity for which of the following. So while I have not, yeah, I did not show this interaction in my discussion today, but yeah, it has been previously discussed quite extensively. So CXCR5 and CXCL13 interact with each other, and it's actually a driving interaction towards the movement of centroblasts from the dark zone to the light zone, and for them to actually see their antigen from the antigen presenting dendritic cell and execute its downstream action. B cell receptors having high antigen affinity are selected in the light zone. This process is known as what? So BCRs or antibodies having high antigenic specificity get selected in the light zone by a certain process. If you remember, there were two substeps within the dark and the light zone, and that was diagrammatically represented by Professor Agneo as well. So the process that takes place within the light zone, he called it as affinity selection or affinity maturation. The more commonly used term is affinity maturation. So that's what we will call here also. Even if you did not remember it correctly, if you look at the other terms, it's quite easy to deduce the correct answer in this case. Obviously, it's not affinity tagging because that is a tag attached to some protein, either an antibody or a protein molecule. So that's obviously not the correct option. It's definitely not recombination because that obviously has nothing to do here as well. So the options here were quite vague, and if you get a question like this, it is quite easy to narrow down to the answer very quickly. Finally, you have the next question for interactions between B cells and TFH cells, which are follicular helper cells, which initiate or induce the process of class switching of B cells. They are as following. So B cells and T follicular cells, the interaction between them is mediated by which of the following? A, MHC2 TCR interaction. Obviously, that's the most important interaction that is helping in recognition of the antigen that the B cell was presenting, and therefore it will, select for such a B cell and will allow it to go for class switching at all. If the B cell antigen is not getting recognized, then obviously it's a futile step to even carry forward. Second, you need to have a co-stimulatory interaction. In this case, that is CD40 ligand interaction, which is option B as well. Therefore, since both are correct, the correct answer in this case is option C. Availability of different cytokines lead to differentiation of T cells into its subtypes. Match the following with the respective T cell subtype. So this was actually discussed by Professor. I did not have it on my slide. So you can go back to the slide. I might have a screenshot attached, but I'll check it. I don't have it here. Just excuse me a moment. So yeah, so in this case, the different type of cytokines that are present that are associated with different T cell subtypes need to be matched with each other. So let's just go by the answer directly so that we can address the question. Option A is the correct option. So let us look at the combinations there. IL-12 is associated with TH1. IL-4 is associated with TH2, and IL-6 along with TGF-beta, transforming growth factor beta, is associated with TH17. So these are the three types of T cells that are differentiating based on the type of cytokine which is available at that particular point of time. IDAMs present on the cytosolic domains of Ig alpha and Ig beta transduce the signal for B cell activation. What are IDAMs? IDAMs are immunoreceptor tyrosine-based activation motifs, or B, option B. I'm just coming to the answer directly. And finally, for the last question, what is the site for activation, proliferation, and differentiation of B cells? A, the germinal center of the lymph node, B, the MALT, C, blood, or D, all of the above. So it is A, germinal center, obviously, because if you remember correctly, we have discussed that quite extensively, that it is the site of active B cell proliferation and differentiation. They are like small uh, little patches which are present towards the outer side of the lymph nodes or the cortex of the lymph nodes, and that is where the entire all of these processes that we talked about, they take place. So with this, we are coming to an end for this class. Since there are no other students today, I will be ending the discussion. And yeah, I hope the course has been interesting for you. I was hoping for better, attendee. I was expecting some attendees to be present, but it's okay. I understand maybe it's difficult, but please try to, participate in these discussions. Even if you can join briefly, that would also be all right. Yeah. So apart from that, thank you for your patience, and all the best.